protein synthesis. Yes, this came a long way. Since grade 8, we have studied the cell and the nucleus of the cell with its important genetic information. Now we know that the genetic information is made of DNA. We also know that DNA contains a specific code. This code is in the sequence of the bases, the nitrogenous bases. And today we are going to see how that code on the DNA is used to make proteins. Protein synthesis comes with the expression of the gene. The sequence of the nitrogen bases will eventually tell us the code of the protein. I would love us to quickly look at proteins. Yes, we know that proteins are made up of a specific sequence of amino acids bound by peptide bonds. This sequence will then be folded into the secondary structure of a protein, which is held together by hydrogen bonds. Then we have the third or the tertiary structure, which make these bonds into a three-dimensional structure. Now this three-dimensional structure is held together by disulfide bonds and also hydrogen bonds. That is the tertiary structure. But proteins also have a quaternary structure. We have different polypeptides making proteins bond together. We need proteins because it makes up living material. We know blood contains hemoglobin. The brain needs the channel proteins in order to get the brain signaling small molecules to enter the nerve cells. We know at the synapses, this channels is important for calcium and sodium ions to enter. Enzymes need to digest inside the mouth, inside the stomach, the small intestine. But enzymes will be all over in the mitochondria. Enzymes will catalyze reactions, all made of proteins. Then we have clusters of proteins to help the cell copying genes and making cell division and new protein synthesis possible. Antibodies. Proteins that helps with the immune system. We call them immunoglobulins. Cell messengers, the receptors at the cell surface membrane that needs to communicate with the inside of the cell. And also hormones are messengers, chemical messengers made out of proteins. Then we have the muscles and the hair and nails. We also divide proteins into globular and fibrous proteins. And just to help us to remember, let's add hemoglobin, globular protein. So it's a spherical shape. The channel proteins, globular. Enzymes, globular protein. Clusters, globular protein. Antibodies or immunoglobulins, globular proteins. The hormones and the receptor proteins outside the cell membrane, globular proteins. And then we have the fibrous proteins, the keratin in the hair and nails, and the hooves, feathers of other organisms, other animals. And then the muscle proteins are also fibrous proteins. Wow! Now we did a lot of protein revision. That is the recipe, dear grade 12s. Repeat, repeat, repeat. Now, we are going to see how these proteins can be synthesized. So, we have a sequence of amino acids to be connected by a peptide bond to one another, and eventually it forms a polypeptide. The polypeptide is folded and bonded together to make a protein molecule. We have 20 different amino acids, and how do we get the amino acids? We get the amino acids by what we eat, right? So, this amino acids will be part of the ribosomes. The ribosomes will take all these different amino acids that we got from what we have eaten, the proteins that we have been eaten, will be digested, and I will have lots of amino acids in my blood. Now the ribosomes will have the job to assemble them into the correct proteins. Where do I find ribosomes? On the rough endoplasmic reticulum, and also lying free in the cytoplasm. If now I am looking at the functional components of protein synthesis, we of course will start with DNA, the double helix with a specific sequence of nitrogenous bases. This nitrogenous bases for the DNA, cytosine, that will always bond with guanine, and adenine, that will bond with thymine. Always bonded like that. It is not a random sequence, but this sequence will hold the key. This will be the code for a specific amino acid. 3 of them. Okay. Then we know that we have RNA, and especially the mRNA needs to link with DNA at a specific stage. So it also have cytosine, which will link with guanine when it needs to link with the DNA. Adenine will now link to uracil. Then we have the RNA. So here we have our DNA molecule. DNA contains cytosine and guanine. Guanine and cytosine are linked in this double helix. We call them complementary strings. They do not look the same. On the one side, if I have cytosine, on the other hand, I will not have cytosine but guanine. Then I have adenine and thymine, which will link together. Okay, then I have an RNA strand, which is a single strand. And here I see, okay, cytosine and guanine resembles DNA, adenine resembles DNA, but uracil is a different nitrogenous base or nucleobase that I find on RNA. So why? To make protein, we need the following to take place. We need transcription. So, transcription means to copy the genetic information from DNA to RNA. We call that transcription. RNA will copy the genetic information, but the copy on the RNA will be the complement of the DNA message. You agree? That we call transcription. Why do we have transcription from DNA? Because DNA contains the genetic code for protein. It is needed to get that message to the ribosomes that is outside the nucleus. But the DNA is large and the double helix cannot leave the nucleus. So therefore, what needs to happen is the DNA needs to unwind, unzip, and to unzip it needs breaking the hydrogen bonds between the nitrogen bases. Now the messenger DNA will come and make a copy. Again, it is not an exact copy, but it will form the complement. It will form the complement of the message. Because the single strand of RNA can leave the nucleus through the nuclear pores. So what will happen is, if we can just go back to our previous slide, the guanine of RNA will link to cytosine. So let's make a note here and to help us to remember. The guanine on the RNA will link to the cytosine on the DNA. Right? There we have RNA, guanine will copy the cytosine. The adenine of the RNA will link to the thymine of the DNA. The uracil on the RNA will link to the adenine. Right. The cytosine on the RNA will link to the guanine on the DNA. I hope you do understand that. So now we have a transcription, a copy of the DNA that opened to have the part with the specific code for a specific protein open. So transcription, a part of the DNA unzips, unwind, unzip, breaking the hydrogen bonds. mRNA comes and take only the part with the code on and copy the code from the DNA, but in a complementary message. It will copy the complement and there will be no thymine but uracil. That is what we call transcription. How does it take place? There are 3 RNA nucleotides inside the nucleus, and that was produced by the nucleolus. Make the RNA, the 3 copies attach in the correct complementary order, and now it is called transcription, leaving the nucleus. The mRNA goes through the pores with the message. It's taking this message to the ribosomes. In the ribosomes, there are transfer RNA. Transfer RNA. Which is, this is a tRNA or a transfer RNA. We see that attached to each transfer RNA is an amino acid. So this amino acid will only be attached to this specific 3 bases. This 3 bases is called the anticodon. This anticodon inside the ribosome will fit 3 specific sequence of a mRNA, and that we call a codon. On the RNA, mRNA, we have a codon. And just to help us to remember that it is 333, they call the 3 there that will belongs together to form a codon to fit a specific anticodon. On the DNA, they say there must be a triplet of codes on the DNA that will form the codon of the mRNA. On the codon of the mRNA, there will be an anticodon. Attached to that anticodon, we have the specific amino acid. And now we call this translation. When the codon is fitted out with an anticodon, now there will be a specific order of amino acids linked together, forming a peptide bond, and that is translation. So, let's quickly talk about translation again. The decoding of the mRNA, 3 bases of mRNA, will attach to a tRNA. The tRNA is like hooking 3 specific nitrogenous bases, because this 3 had that amino acid attached to it. As this one is leaving, a next one can attach in the ribosome. And there I will then have a specific sequence of amino acids which will form a polypeptide string. That is translation. The mRNA coding for a specific tRNA with its attached amino acid that will be sequenced inside the ribosome. That will be called translation. The transfer tRNA carries amino acid from the cytoplasm to the ribosome. It will attach to a specific 3. These specific 3 will be called a codon of mRNA attached to an anticodon on the tRNA. So where did this amino acids came from that is now in the cytoplasm? That came from the proteins that I ate. It got digested or hydrolyzed inside my digestive system, absorbed by the blood, transported to my cells, and now the amino acids can get into the cells. This tRNA, together with the code on the mRNA, rearrange all the amino acid so that I can have a new protein with a specific function in the human or any other animal or plant cell. And again, a series of 3 adjacent bases on the mRNA is a codon, and a series of 3 nucleotides that are complementary to the codon will be on the tRNA. What will be the complement of this codon? G will have a C, C will have a G, A will have a U, because we are talking, and this will all be attaching to a specific amino acid on the tRNA. Right, so each tRNA codes for a different amino acid. Messenger RNA do transcription. mRNA is transcribed in the nucleus. The mRNA can leave the nucleus through the nucleopores because it is small enough. It moves through the cytoplasm and joins the ribosomes. Inside the ribosomes, which is the factory of proteins, the codon of the mRNA will be translated by tRNA. The mRNA enters the cytoplasm attached to a ribosome. Translation begins at AUG, which is a start codon. Each transfer RNA, tRNA, has an anticodon whose bases are complementary to a codon onto the mRNA strand. The ribosome positions the start codon to attract its anticodon, which is part of the tRNA, which binds methionine. The ribosome also binds to the next codon and its anticodon. And then I have an assembly line of amino acids joined by a peptide bond to form a polypeptide. When I have a polypeptide and it completes its 3-dimensional structure, I have a protein. So we can use a codon chart, which is fortunately set by all the clever scientists that researched protein synthesis, and now we can discover which amino acids will fit. So when I have CAC, I look for C in the first base, A, and here I have a C, right? Third base. So it will be histidine. You agree? You can pause the video and see if you can find another one yourself. C. Again, C. The second one is C and an A. C, 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 A. There it is. So it will be proline. U. Let me use another color so that we do not get confused. U, G, U, G, A, U, G, A. UGG, first UGG. UGG is tryptophan, and then I have a stop in UGA. So, you got that? Right, then the next one is a different type of chart to help us, and we start in the middle. A-U-G. Aha. So the first one will be methionine. Again, you can try and find them on your own, and then play the video again to see. There we see methionine, asparagine, aspartic acid, and then a stop. To capture this, we have DNA needs transcription by the mRNA to get the code of the gene to the mRNA. mRNA will be translated by tRNA, and that will form the protein. Add your own sounds. RNA polymerase. Doing the unzipping. RNA subunits produced by nucleolus. So I added a quick revision sheet for you so that you can quickly see and bind the correct names in your pathways in your brain, dear class. Please print this one as well as the added summary that also will help you to quickly revise it and the worksheet attached. Please try your utmost best. I think that you then repeated it enough, and if there are any more things that is not clear to you, just watch it again or read through your textbook